And now our last speaker of the session, Dr. Lucio Freitas-Junior. Professor? No, I think this was a mistake in the. Lucio, following his PhD in Sergio Schenkman's lab in Brazil, moved also to Institut Pasteur France, where he's been working on malaria. And he also did a small tuition stop in Brazil before moving to Korea, Institut Pasteur Korea, where he was instrumental under the DNDi mandate to set up the first high throughput screen for intracellular mastigote leishmania in 384 well-plate format that led to the development of other assays in other parts of the world using the same imaging system or similar imaging system. Now he's moved back to Brazil, trying to set up a discovery platform, working on different parasites. And today his talk will be about T. cruzi and the challenges with this parasite. Lucio.
Thank you very much, Eric, for the introduction, and thank you for the invitation. I must say that I'm extremely relaxed. I mean, here, I don't know if it's too much wine that I got in the lunch. Just because I came back from my house. I mean, I was here for 5 years. And I mean, I spent a very good time here. And as Eric said, I mean, now you implemented a center for drug discovery focused on neglect disease in Brazil. I'm going to talk specifically about the place later. So I'm going to discuss today the new challenges involved Trypanosoma cruzi, I mean, Chagas disease, drug development. So I play in Brazil that Chagas is one of the main exports that you have in Brazil. You know, you share with everybody now. I'm not jealous about that. So I'm not going details in the life cycle. I mean, but just to point for you guys that there's something there is so much to be done, so much to be studied in Chagas disease. It's a paradise for basic research, but for drug development, it's a nightmare. I mean, you have a very complex life cycle. I mean, when the parasite undergo a differentiation to infect cells, so this parasite can infect any cell in your body. This is the danger. So if I need to bet my job on the whole cell that I'm using today, I will not bet. I mean, it's something that I also need to see, you know, and address this later. But anyway, after the infection, you have the acute phase, very short, and you don't get problems, serious problems in this time, but the problems come 20 years later, 30 years later. Some of the patients, I mean, develop some megaesophagus, megacolon, and cardiopathology. I have a friend that is in the hospital right now, I mean, with megacolon in Brazil. This has happened. And so we have two problems here. So you have infections, so you need to find drugs for acute phase, but also you have many people infected that go to get this pathology expressed in 20 years for later. There's something also that you need to be worried about this. So you need to develop compound drugs that can interfere with this process. So with DNDi, you did together a screening for Leishmania donovani using the human macrophage. So since I have a background on cruzi, we began to explore, to do some, to apply the same technology in Trypanosoma cruzi. So very quick, I mean, you set up a protocol, a SOP for infection for Trypanosoma cruzi. You have a software that was developed by Augusto Genovese that is here today with us. That was a big collaborator, I mean, with our group. So this is what the expertise that you bring for the that Institut Pasteur did, you know. So they bring this complexity of image for the drug discovery. You can get so much information from an image like that. I'm going to explain this to you guys later. So you did the screening you set up a protocol. So the software was designed. The software is very similar with Leishmania donovani, but I can tell you there is no bad day for cruzi. Leishmania is difficult to control. We have bad days, good days. Cruzi, no bad days. So it's a system that is very difficult to control. Of course, you have the risk of infection. I have friends that when I was doing PhD in Brazil back in 20 years, I mean, I have people that got infected, so we need to be very careful dealing with these parasites. And very quick, I mean, you have done a screening. First, I had to put the screening for phenotypic. And here you have these compounds in red. Most of the compounds have no activity. You have bezinidazole here, okay, in yellow. And you have many hits, so you got potential many hits there. the question is, I mean, it was so easy to do this. It was so easy to do that. So for Leishmania, it took us 9 months to set up the protocol. For cruzi, it took us 2 months, you know, and it was very quick, so quick that, I mean, you began to think. I began to read about drug discovery in Trypanosoma cruzi, I mean, and I found a black box, you know. There is so many questions that I go to discuss with you guys that you need to take in consideration, that you need to share in order to have a competitive drug discovery. For example, genetic diversity in Trypanosoma cruzi strains. So Trypanosoma cruzi was a work that was done in the 80s by Michael Myers for the London School. So he classified Trypanosoma cruzi in 6 big strain groups, very diverse. I have worked with Trypanosoma cruzi on my PhD, and I knew that they are very different between them, you know. So one strain is completely different from the other. The question is, the strains that you have done in the screening in Korea was representative for the other groups? So this is a new, now you have a new classification that was published by Professor Bianca Zingales from São Paulo, from USP. And you can see the 6 different groups of strains of cruzi. I use this strain, the strain that comes from this group here, Y strain, okay? I just choose this one because it was a strain that infected very well. It make our life easier. Doesn't mean that you go to translate later for something, you know? So sometimes you make shortcuts. But you need to understand that the shortcuts that you get in the beginning can compromise our research later. So the question is, how representative is this strain here? So DNDi also have the same question, I mean, and so in DNDi then ask us to develop assays for all these strains. So we got in contact with Professor Michael Myers in London, and he sent for us representative of these strains. So we got two examples for each one of these groups. So it took us 1 year to have all these strains running. It's very difficult. Some of these strains, when you look in the lab, some of these parasites, they are very different one from the other. You know, so it took 1 year to, you know, I almost say the word teach. See, this is already biased. You are teaching the parasite how to infect cells. But, you know, the question is, it took a long time to have all this system here running and infect the same host cell. Otherwise, you cannot compare the data. And look at this. So bezinidazole is the drug that people use today to treat patients with Chagas. And each color you have here, you have a different group here from one different group of different strains. See, for bezinidazole, they behave in a similar way. So you have the parasites clean, you have the infected cells clean from parasites, I mean, 100% of the case, okay? EDIC 50, very similar. And here's posaconazole that fail in clinical trials. So just to tell you how important can be, I say that to develop in the lab. So it took us 1 year. And to be honest with you, I didn't expect to see a result like that. But when you compare the posaconazole that so far have, you know, I go to show some data later, was very promising. In our assay, they fail. You see, they can kill some of the strains, very efficient, but not all the other strains. So this assay, this secondary assay is an assay that is extremely important. If you want to go forward, you should use these spin-off strains. Just a word already, these spin-off strains I have available with me in Brazil. So if you guys want to test compounds, if you guys want to test anything, we can test for you guys, no problem. We are very open for collaboration, very open. You did this also for Newfortimox, that is used, again, Chagas, to treat patients. You see, similar. But you test other compounds, like this one, I mean, like this compound that is in clinical trial for sleep sickness. And you see, and lead optimization for Chagas, and some of the strains fail here, okay? Havaconazole, the same class of the posaconazole, fail in a very similar way. You know, this was also failing in the clinical trials. It is last 2 compounds that belong to the same class as a herbicide and was in the lead optimization in the DNDi. So you test this, this is a panorama, you know, so it's very complex. So this assay that has become so essential for us I think is essential for drug discovery for Chagas disease. And a new concept that you have, because when you see bezinidazole is in the range of micromolar here. Posaconazole was so promising, nanomolar. So when you're looking for drugs, you go for potency, right? So you need to look for the potency. But this is not so important because bezinidazole is very efficient, and posaconazole is very promising, and some of the strains they're not efficient. In modern days, if posaconazole don't clean the cell curve, they infect cells from the parasites. There is always parasite that remain there. They are not very efficient. So you decide so when you see here, so you have infect cells over 6 days. So in different concentrations of bezinidazole, posaconazole. So once you make sure that posaconazole was not working because maybe you have a short time incubation. So you left them for up to 6 days, and doesn't matter the concentration that you put. They don't clean all the parasites there. So if you cannot clean the parasite, there is a chance that you go to have parasite that become resistant. So a fast kill is also something important to take in consideration for drug discovery for Chagas, I would say. I'm not saying potency is the best, it's the more important thing, but efficient to clean and quick, I would say, is important. So in Korea, then, our expertise was then phenotypic screening. So, and I believe this is the best system so far. So the idea is using, libraries in this system, complex, trying to get the system as close as possible with the disease. Never going to be close enough. But the question is, you're going to get hits here, and soon as possible, this, for us now in our lab, you want to test this compound that comes from this, right away in the span of strains that you have. Because it doesn't matter, if you cannot cover all the strains, it makes no sense to follow. And only after you have like time kill, mechanism of action, and using another parasite that you have in the lab is also important. So chemistry for us only start after you test the pine of strains. So you don't want to spend money and time in something that is not important for the whole strains. So the first item I just discussed, the diversity, and we are ready to share this kind of strains with you guys. But also something very important is the biology of the parasite. So this is a very elegant, if I can say, parasite. So you have, for example, Leishmania, once they infect the human host, they keep as a mastigote there. Trypanosoma cruzi, they undergo a very extreme differentiation, okay? So you have a triple mastigote that is a machine that is made to infect cells. They need to infect the cells quick. They infect the cells quick. By the way, this was done in viral cells, just for the record. So they infect the cell here, and once they infect the cell, they need to become this form that called a mastigote. So this is the form they multiply inside of the host. And once in this form, a mastigote, they keep dividing, and the cell, you see the cell, don't see the parasite inside. The cell, with parasite, they should commit apoptosis, but the parasite can manipulate the cell. The cell is so happy here, they go in the division here, cell division. So they cannot see the parasites inside. So the parasite is really, controlling the whole thing. They want to have the cell happy there, synthesize amino acids for them, so the parasite needs this. And after the cell divides, you have parasites here, you have parasites here. And this process goes as a mastigote until the cell is full. So see, the flagellum is not beating too much, but the parasite, you have no more space, it's time to go. They begin to differentiate back in this triple mastigote. So the flagellum elongate again, they get even more elongated, and just in the right time, they go to infect new cells. So again, so complex this. So maybe you can try to find drugs that can block this process here. So differentiation is very complex. I will stimulate something like 600 proteins involved on this complex pathway. And you see, so when I was back in Korea, work with August, and August asked for the biology, So Lucio, tell me what is a good hit? What is a good compound for Chagas? And I told him, so this is a bad one. So most of the cells are infected. So have 90% infection of the cells. Most of the cells are infected with many parasites. And I told him, so this is a good one. few cells infect, you know, so 5%. So when you did the whole screening base, you know, think about that. But after you want to go back on this idea to find compounds that can block the parasite. Imagine, the parasite infect the cell as a triple mastigote. Just imagine that you can find a compound that can block the parasite as a triple mastigote inside of the cell. So you need to do the whole screening again. Not really. You just get the same image that you got already before, and he wrote the software not to give the less infect, but very infect, most of the cells infect, but with few parasites inside. What did you get? What do you get with that? You get a completely set of different hits. See, I mean, there's something that you need to explore in drug discovery, I mean, to see for the weakness for your system. This is the classical graph for drug discovery. Positive control, the MSR, most of the hits don't interfere. I did a proof of concept with 4,000 compounds here. Okay, it was a library from Biofocus. Focus was a library against kinase. Just, I don't say that as a good as a target, as a drug, just I use what I have there. Most of the hits don't interfere, and you have some good promising hits here, near to bezinidazole, the blue one. But the software, and you ask them to give one parasite for cell, many cells infect, they give these pink hits here. This no one that works with screening, you're looking for these ones here, right? So you got this, and guess what you got there. So you have here the host cell. This as a coloration for, this is a staining for DNA for the host cell in red, okay? And here in green, I have antibody against the replicative form of cruzi, against a mastigote. So if the mastigote is there, you have this green signal. So I left 12 hours. You have one parasite here for cell. So you see it's very strong. So the parasite is happy, multiply, very strong. In the 72 hours, there is no more space. The parasite needs to differentiate for the infective form. In the infective form, you don't have the antibody, you don't have the epitope for a mastigote. So there is no signal here. Okay? So bezinidazole, this is true for bezinidazole. Okay, the parasite is very weak, so you see the parasite is inside, but the epitope is weak there. So the antibody don't see much. Why? Because the parasite is dying. The parasite is not happy at all there. But look for this one here. So look at that. As I told you guys, I mean, there is many people infect out there. So you have chronic patients infected with Chagas. You don't find a parasite in the blood, but you find nests of mastigotes in tissues, like heart, many tissues there. You find nests of a parasite sleeping, let's say. It's not clear yet if the parasite is sleeping, what is going on, but you find these nests there. So there is many mastigote nests. There is no parasite circulating. So in here, you think that you find a compound that can induce the parasite to sleep. The parasite is not die, it's not dead. See, because the antigen is very strong, it's very green. And I keep 2 parasites for cell up to 72 hours. So our idea now, this was not the idea, you didn't expect for this result there. So, but now, maybe you have a system that you can induce for that format. After this format is induced, I bring new libraries to see what can kill this. Because kill something that is replicating is easy. But kill something that is sleeping there is going to be a challenge. So this is a prototype that I'm working, you know, so. But I was looking for this one here. So in the last hit, what I have is difficult to see. I mean, the light is not helping, but you have host cells, and you have one single parasite for cell. So you can see the DNA here is very weak. So, but this parasite never got the antigen for the mastigote, never. So we have a slight chance that maybe the parasite got as a triple, and it stay as a triple. So what you did, I got a triple specific antibody to test this. So see, triple specific antibody, they don't cross-react in the beginning. You have only a mastigote, only 72 hours, when the parasite began to differentiate back to the infective form, you begin to see these antigens there. Okay? Bezinidazole, nothing, and you look for this here. You have here triple mastigote specific antibodies, cross-react with one single parasite for cell up to 82 hours. I'm not saying that's going to be a drug, but can give us a lot of insight about the biology. So this was the goal from the beginning. So this is the nest of parasites that you see in heart tissues. These patients that die with Chagas, with cardiopathology, okay? And you see this parasite is sleeping there. So maybe with that compound that I showed to you guys, you have a window to explore this, to develop assays against for chronic patients, okay? So for the last, but not least, as I said, you have very good science for Chagas, very good Chagas. You have Paulo Minopra here that published a Nature a few years ago here in Pasteur with Ramanais for Chagas. But you need to, back in Korea, you have to find a way to find new targets, but targets that can be drugs. So cannot be enzymes that are work already. It need to be cannot be bias system. And you just published this paper here with Augusto that he's in the audience. So you develop a technology that you can knock down every single RNA from the host cell, every single RNA that you encode for a protein. So you have done this. So the idea was to knock down every single protein from the parasite, from the host cell, every single one, the 25,000, 21,000. And after infect the Trypanosoma cruzi to see when the parasite go to have problems to develop. Okay, so this was the validation of this technology. So this was a really breakthrough in this field because these RNA experiments that you did this in arrays, very small arrays like this one. So this is the slides that you have in the lab. So in your single slides like this group that was Augusto's group and Neil Eman's group, they developed this technology. They are able to spot 4,000 host genes. So in every single slide, you have 4,000 human genes divided in the space like this one. So here we have one gene, here you have another one. Here, for example, you have a host cell expressing GFP, okay? And here you have knockdown for GFP. You see, when they have the spots, no GFP expression. So you have you have set up this protocol for cruzi that you have already the slides with the RNAi. You see the cell, okay, you let the cells go transfect for 48 hours to make sure that the cell have no more that specific protein. Many of these proteins that you did knock down was toxic because the cell need that protein, okay? But you need to take this in consideration. And after 48 hours, you infect these cells with cruzi. And Augusto then developed a software specific for that took in consideration many factors like cell number for every single spot. And you come out with a list of hits, okay? You come out with a list of targets. I did this in Y strain, okay? So I not say that you have validated targets here, but some targets at least in Y strain that the cruzi cannot develop if the cell don't have this protein inside. Of course, we did this with Y strain. You need to validate this in other strains, but it's a way to have a system unbiased, putting light in mechanisms that you even don't know that exist. And another way also that you using this technology by image, something that you have done, you did this with DNDi, you have done this with DNDi, because as I said to you guys, our system is cell-based. You don't care about the targets. So in fact, the cell with parasites, you test our compounds, and you get the best hits there that can kill the parasite inside of the cell. Don't interfere with the host cell. There's something nice about phenotypic screening. You can have already idea about toxicity when you're doing the primary screening, okay? And once you get these hits, it's already good to go. But with this image technology, you can add different questions. For example, this is an organelle that you find in these parasites that belong to the Kinetoplastid, okay? Like Leishmania, cruzi, sleep sickness. All these parasites, they're very similar. You have the nuclein, and you have this kinetoplast that is a modified mitochondria, but with a very specific role. Okay, this mitochondria, these kinetoplasts have up to 500 proteins there, okay? And every time that you're trying to knock down one of these proteins here, the parasite die, every single time. So this organelle is essential for the parasite, essential. And when you compare Leishmania, brucei, cruzi, just the same. They are very similar. You find one protein in one, they are present in the other parasite. So the idea was, you have done the screening for Leishmania with DNDi back in 2009. So you have done 2,000 compounds in the intracellular model for Leishmania. So you choose the best 2,000 compounds, and you did a secondary assay with this. The idea was to test, again, the same compounds that come from the intracellular model and testing 4 different parasites. Leishmania again, Trypanosoma brucei, and cruzi. Why these 3? Because these 3 parasites, they have the functional kinetoplast. They have the kinetoplast very complex, okay, very similar. But Trypanosoma evansi, that is a parasite that infects horses, they don't have the kinetoplast, so they have a very primitive kinetoplast. So the idea was, maybe you can find compounds that can kill all these 3, but don't kill evansi. There is a possibility that some of these compounds that don't kill evansi, they are specific for the kinetoplast. So from 2,000 compounds, you select then 12. 12 compounds that can kill the 3, but not evansi, don't kill at all. So you have done then a third screening now, set up in a replication assay. So this is a PhD project in my lab, a Korean student, Kyun-san. So the idea was to get these 12 hits that you got there, that you thought that was specific for the kinetoplast, for this organelle, and grow the parasites in the presence of these drugs, plus nucleotides, that every time the DNA replicates, you can see the new DNA. So this is a healthy parasite, so we have replication in the nuclei, we have replication in the kinetoplast. Look at this. You have done, this is positive control, we have replication in both, we have DNA here, and we have replication in both. And you got these 3 compounds. Look what this compound can do, the compound number 1. So the cell keep dividing the nuclei, but the kinetoplast is there, but there is no replication. You see? By image, you can extract so much information. The number 3 compound is like ethyl bromides. You have replication in the nuclei, but the kinetoplast is dead. Okay, just like ethyl bromide. So this is a DNA intercalator. You know already, just like ethyl bromide. And this is very interesting because, as I said, you guys, you did a profiling assay to find something specific for the kinetoplast. You got the luck to get something that visually, you see there's something wrong there with this organelle. But you go even further. By image, you can see that this pattern that you have here, this ring, remains as a ring. It's very typical for DNA, for helicase. Have shown already for Trypanosoma brucei, when you do RNAi for some helicases, the parasite die, you get absolutely the same phenotype. So by image, you can even define the target, so it's very powerful. And this gives for us, the possibility to work in that target that is pretty much, you don't expect to have a cross-reaction in human. You don't have kinetoplast in human. So this is what you can offer now in Brazil. You have up to, not only Chagas, but the other models. Yeah, it's done. And all this work was done then at the Institut Pasteur Korea. So I was there for 8 years, working very close with the DNDi since 2006. It was a wonderful experience, for me. So I come from a basic research background. And there, I need to forget about to do papers. The important was not the paper, and to get a product. And the DNDi was very important for our group, because the Koreans, for many years, I love the Koreans, but see, what you are doing here, we cannot make money with you, But after they saw, that you can do more than money. You can do more than money. You can make the difference, For example, for Korea, it's extremely important. Africa is extremely important for Korea. It's very strategic. And our group become important there because they see on this, they want to show to people they are making the difference, And we didn't spend much. Our group, it was not an expensive group, but it was a very important experience. And now, since March this year, I moved for this new place. So the Brazilian government is trying to improve science in Brazil. We have already very good science in Brazil, but trying to improve the management. So the Minister of Science and Technology, they gave every year a lot of money for this facility. And in this facility, you find the synchrotron, nanobiology. Our group, so our group is in the biology here. You have the institute that is focused on ethanol. So once you develop the second generation ethanol, you're going to take ethanol from the cana bagasse. And our group is focused here. So you are building now a new synchrotron that will be really much larger than this one. And this place is great, I mean, because I don't have fixed position there, just like in Korea. I can lose my job anytime. But they're really much focused on deliverables. They want to see something different. So they agreed to bring, I'm not coming alone from Korea, so they allow me to bring my group. So the data that I showed to you guys today was done in Carolina's Moraes group. So she is the leader. She was with me in Korea for many years. Now she is back in Brazil, coordinated the Chagas team, and with John, that was responsible for dengue chikungunya, that is back here. So I want to say also, of course, thank you for Korea, specific for Augusto, that was a big collaborator that I have there. You did so much together and want to do more. Something that was great, this work. I learned so much there, you know, to be in this environment, very focused is very good. But you have a backside. The backside is, I mean, the Koreans, they want to protect intellectual property is very important for them. So something that, you know, was difficult for us is to have tools like this software. You want to share the software with the people, but are not allowed to do that. It's something that I'm working with Augusto now. We're going to develop new softwares. You know, you want to share this with people, you know. So I think you can hold the ground much better if you can share. There is nothing more old than today's technology. So if I share with you guys, for sure work with something else. And of course, thank you for the DNDi, for FP7. So I have the luck to be in this consortium that Gerard was the coordinator. And I just got another FP7 with Paola Costa that should be here. And Professor Ana Cordeiro, that, I mean, as you have done this work on cruzi together. So thank you very much.
So thank you, Lucio. I think you showed some consensus with our colleague from DNDi about the need for secondary assays and what you can gain from better knowledge of the biology of the parasites. You gave us a nice overview of the different approaches to identify potential targets and to help us better understand the parasite. Any question in the audience before the coffee break? Yeah, Julio?
Lucio, obrigado. It's a great presentation. I just wanted to ask you a question and make a comment concerning those experiments that you showed concerning the high variability among different strains from different DTUs of cruzi using CYP51 inhibitors. The preliminary question is, after I know how complicated it is to adapt those strains, did you find that the generation time of all the strains was the same?
For duplication, you mean?
Yes, generation.
You are doing this right now?
No.
You are doing this now because I think they don't multiply in the same rate.
Exactly,
this for sure.
Exactly.
This for sure.
That's exactly the point.
Yeah.
So in our experience, we have done similar things. The generation time can vary from 24 to 60 hours, depending on the strain, in intracellular mastigotes. So the question is, when you did the experiment with posaconazole or ravoconazole, did you use the same incubation time? And how long was that?
You try different ones. I mean, for us, it's good
when you come in those graphs, what was the incubation time?
You try up. It was the whole thing, the whole 144 hours.
Because the fact is that due to the mechanism of action, if you're using ergosterol biosynthesis inhibitors, you have to wait for 4 generation times to observe the full depletion of the intracellular ergosterol content. And that means that you will have to incubate, depending on the generation time, from 96 to 240 hours. So you cannot compare strains for that mechanism of action with the same incubation time. See what I mean?
I agree. Relation to the duplication time, I can tell you, for example, a Y strain is 8 hours. This I know already. They multiply extremely quick. Every 8 hours they duplicate it, you know. For the incubation, yes. Very similar with epimash goods. Some labs get 10 hours. Some lab get 10 hours, you know. I mean, I was very cautious to don't set up something new there. You know, I have invited Sergio Schenkman, that I was working for a long time with Cruzi. I invite Sergio to come to Korea, set up the system, you know. But I understand your point. I mean, it's difficult to say. I mean, the question is, if you have to incubate the compound for so long, you know, something, you open door also for resistance, you know. I mean,
What we have seen is that you wait the 4-generation time and the efficacy is 100%.
you didn't get this there. And for me, it was very, I put a lot of pressure on my postdocs there, because I keep telling for them, so there's a Julio Rubini in the other side, you guys need to kill this. If they try, you know, they really try so hard, I mean, and not, you didn't manage to.
Part of the natural variability of Lucio. You have to keep your friend.
Yeah, I think the story of the efficacy of or not of posaconazole or azole in general will still need some additional work to really be certain that they behave as they behave. I think what is sure and is that there are so many CYP51 inhibitors around that we don't need new ones. I think, and from a discovery perspective, we have to look for other class of compounds.
Yeah.
Then there are 2 clinical trials that showed limited or no efficacy, so it's a bit worrying. And we start to have preliminary data in vitro that give a starting explanation. Maybe it's not the right explanation. Maybe depending on the state, depending on the lab, depending on the strains, you might have to incubate 2,000 hours to see an effect. But for the time being, that's the data we have, and we have to live with them. But I think it still need, there will still be a lot of basic research around this issue.
The clinical results have to be analyzed as clinical results. That has nothing to do with in vitro results.
Right, next questions? Yeah.
In the presentations, both yours and the ones before, there was a lot of care that went into setting up the in vitro assay, either with extended cultures or with infected macrophages. There was also a lot of care in, you know, measuring the effects and breaking down the type of effects as you described in your presentation. There was not a lot of discussion about the source of the compounds that are being thrown in these assays, and the outcome of these screens is going to be based in part on the quality of the material that's thrown in terms of chemical diversity, in terms of the filters that are put in place for you know, reactive compounds, microacceptors, solubility in organic solvents, natural molecules, and so on. I mean, as DNDi thought about, in fact, standardizing the numbers and the qualities and the origins of these compounds, so that the comparisons between microorganisms and the potency in one organism versus another, versus a clinical trial versus an in vitro assay, can be standardized across some of these parasites.
So, in our company, you have our own library. So you have this library, you know, take care for people that, I mean, you know, you have people specialized on that. And also I can tell you that, I mean, back in Korea, you have tested, I think was something close, more than 500,000 compounds from different pharma. So you work with pharma like GSK, Novartis, all they send the compounds for us. I mean, you know, with quality control, and they send us for us to test in Korea. They confed the data because they put controls there. It was a blind assay, you know, and there's something that is important to take in consideration. I agree with you.
I think the quality of compound or libraries will be the subject of session 2. And maybe what we are trying to do is also a quality control plate with a given set of compound to try to standardize the assay around different labs. So Julio, very
Yeah.
short question and then
Yeah,
coffee
it's-
break?
It's about the dormant stage of or sleepy, as you said, of the T. cruzi,
Yeah.
and whether or not we would be able to test this in vitro. So, I mean, after so many years working with T. cruzi, have you found some way of inducing this stage? I mean, stopping the multiplication of the life cycle inside the host cell with some host cell, with some factor that you could add.
No.
So.
I think you'll be never able to do this in culture, because there you have one single cell. I think the parasite began to sleep when the environment is not easy for him. For example, you have already an immune response very strong. So the parasite need to get some strategy to escape, to stay dormant. You know, I think in culture will be impossible, but with a single cell. But I'm very confident about this compound there. You know. So this compound is put this parasite in this state, like sleeping or something, and you can, after top of that, bring new compounds to trying to break this. But in culture, I think it will be impossible.
Okay, I would like to thank the audience for all the questions and the attention, and the speakers again for really nice talks. And now it's for the coffee break.